Hello everyone. Myself Dr. Prasad Jamkhande, and today I am representing PATE 2022 pharmacy paper analysis of SRTM University of SRTM University. Uh, I want to introduce myself. My basic qualification is from pharmacology, and I have completed my PhD in anti-angiogenic activity. Currently, I am working as a professor to Shri Ganesh Ramchandra Khatrowar College of Pharmacy, Manwat. With this brief introduction, I would also like to tell you that my research profile or my Google Scholar includes more than 40 different national and international research papers, having more than 2,400 citations. With this little introduction, I would like to start the paper analysis. Hope it will be helpful for you. I also, before starting, I also now would like to thanks Dr. Gingine sir for giving me opportunity to present this paper analysis on this particular platform. Okay, thank you. Let's start the paper analysis. There are some of the general instructions which our university has mentioned before solving the paper. Actually, there are 50 questions are involved, and you have 60 minutes of time. There is no negative mark system in PATE examination. Remaining instructions are the general instructions. Question number one, the determination of vapor concentration and carrying capacity of the gas is termed as, which is the particular terminology used for the measuring vapor concentration and carrying capacity. And the answer is the psychrometry, because drying is nothing but the removal of liquid from content. Evaporation is nothing but the conversion into the gaseous form. Non-thermal drying is also technique of drying where direct heat is not used. But for vapor concentration and the carrying capacity of gas, psychrometry is the correct term used. Next, revalidation in developmental analytical procedure may be necessary in the following circumstances as per ICH guideline. As per ICH guideline, what are the different circumstances where revalidation is needed? Example, first option, change in the substance of the drug substance. No doubt, in this particular condition, revalidation is required. Change in the composition of finished product. If finished product composition is changed, then again revalidation is needed. That means this is also correct. Change in the analytical procedure. Due to any reason, if we change the analytical procedure, then revalidation is mandatory. So the correct answer is the D option. In all these conditions, revalidation is needed. One or a few constituents specific to particular herbal drug responsible for claimed activity. Example, curcumin for turmeric, identify the uh, a type of marker. Particularly for herbal drug or herbal chemical, which particular marker we are using. This is related with the uh, chromatography. We use chemical markers, general markers, or active principles type of markers. No doubt, we are using the active principle type of marker so as to identify uh, herbal chemicals. That's why correct answer is the C option. Colloidal colloidal gel barrier GRDTS is which type? Actually, GRDDS, that is nothing but the gastro-retentive drug delivery system. And this gastro-retentive drug delivery system, particularly colloidal gel barrier GRDDS, is non-effervescent type. Effervescent type system contains the gas forming agents or the volatile liquids, and because of which there is the floating occurs in case of the effervescent. But in case of the non-effervescent, there is no such gases or volatile liquids causing the floating. So this particular GRDTS is the non-effervescent type. In case of the bioadhesive, there is just a binding occurs. So the correct answer is the B option. All of the following statements regarding bioavailability of the drug are true, except, actually I have mentioned the expert, the correction is here, except. Regarding bioavailability, all the statements are true. We have to identify the wrong statement. It is a fraction of administered drug that reaches the systemic circulation in unchanged form. No doubt, whenever we take the drug, some of the amount of drug reaches in the blood in unchanged form. That is nothing but the systemic circulation. So this is the correct statement. Bioavailability of an orally administered drug can be calculated by comparing the area under the curve after oral and intravenous administration. AUC, area under the curve, is used for determination of bioavailability. That means it is also the correct statement. Low poor bioavailability always and necessarily means the poor absorption. This is the wrong statement. Bioavailability can be determined from plasma concentration and urinary excretion data. No doubt, some of the traces also removed into the urine. That's why urine analysis also we are performing to get the quantity of the drug. So the correct answer or the false statement in all these four statements is the C option, which is nothing but the low poor bioavailability always and necessarily means the poor absorption. This is the wrong statement. Mechanism of action of polymixing is based on following one of the classification. Polymixin is your antibiotic used for for the treatment of systemic infection of multi-resistance organisms. Some of the microorganisms are very dangerous. Example is the Pseudomonas aeruginosa infection. And this polymyxin is the antibiotic drug, and it produces its action by directly acting on the cell membrane and affecting its permeability, which is ultimately responsible for the leakage of intracellular compounds. That means this particular B statement represents the correct mechanism of polymyxin. That's why correct answer is the B option. Whereas the A statement represents agent that inhibits the synthesis of bacterial cell wall. It is correct, but it is incomplete. Agent that affect the functions of 30S and 80S ribosomal subunits to cause reversible inhibition of protein. It is wrong mechanism. It is the agent that affect the nucleic acid metabolism. It is also wrong. It particularly affects the cell membrane. Absorption arising from CS stretching from alkane occurs in general region of, particularly they have mentioned the four options representing the different wavelength region. CS stretching particularly occurs in 3000 to 2840 per centimeter, particularly this area. So the correct answer is the C option. This is a very straightforward kind of question. You know the correct values. In India, which schedule covers the minimum requirement of manufacture and quality control of herbal medicine product under the Drug and Cosmetic Act? For the particularly manufacturing practices or good for GMP, good manufacturing practices for Ayurveda, Siddha, and Unani, it is mentioned in Schedule T. And this Schedule T particularly is related with the herbal medicine. That's the correct answer is the Schedule T. It is also straightforward question. How many structural isomers are responsible for C4H2Br? We know that isomers is nothing but molecules with the same molecular formula, but their atoms might have the different arrangement or bonds. So in case of this particular structure, how many isomers are possible? That is almost four different isomers might be possible. Next question. Dash reaction, which constitute approximately 80% of adverse drug reaction, are usually a consequence of drug's primary pharmacological effect. Example, bleeding because of from warfarin or a low therapeutic index. Example, nausea because of the decoxil. And they are therefore practicable. This type of drug belongs to which particular category? And the correct answer is the type A. Actually, this type of drug is according to a degree of overall harm from misuse of the drug. And this type represents, or this type is based on the United Nations Single Convention 1971 and misuse of drug at 1971. That means by using this act, they have categorized the drug in the different types. And this type, particularly this statement represents the drug of type A. Next, Which of the one is not an official test for tablet in Indian pharmacopoeia? Indian pharmacopoeia gives the official test for the validation or so as to evaluate the different dosage form. Example, in case of the tablet, uniformity, disintegration, and content uniformity are the official test, whereas the hardness is not the official test. Actually, official test helps to determine the physical, chemical, mechanical, and biological property of the different dosage form. So the correct answer is the second option, hardness. For the solid oral dosage form, A is generally at a minimum one-tenth that of full production scale, or 1 lakh tablets or ever the larger. Okay, this particular condition represents the which particular batch or scale. In case of the primary scale, we produce a very less number of the dosage form. Whereas in case of the pilot scale, we produce the one-tenth on the scale, sorry, one-tenth that of the full production. That means this one-tenth represents or 1 lakh tablet represents the pilot scale production. Whereas in case of the production batch, large number of dosage form or the tablet is produced. So the correct answer is the B option. In case of the R&D, also required amount of dosage form is produced. So the R&D batch is also the wrong answer. So correct answer is the B option. Next, question number 13. Metoprolol GPCR, G protein coupled receptor therapeutic target is. In case of the metoprolol, which particular target? That means it is also mechanism based question. Metoprolol is used for the treatment of cardiovascular complications. Example, to reduce the blood pressure, to minimize the heart attack. It is also used in case to treat the angina. And it particularly antagonize the uh, uh, 1AR receptor. That means it reduces the blood pressure by simply uh, acting on the adrenergic receptor or inhibiting the adrenergic receptor. Next, cyclopentano per hydrophenanthrin CPPP ring is present in particularly which uh, nucleus this type of ring is present, they have mentioned. Number one is the steroidal nucleus. Second one is the anti-cancer third one is the anti-neoplastic drug, and fourth option is the none of the above. In case of the steroidal nucleus, this particular ring is present, CPPP, which is also called as a guanine, which consists of the four hydrocarbon ring fused together. So the correct answer is the steroidal nucleus. Next, Shinoda test is used for determination of number one glycoside, second one terpenoid, third one flavonoid, and fourth option is the tannin. We know that in Shinoda test, extract along with ethanol is mixed with magnesium and hydrochloric acid, which gives the dark cherry red color. And this particular cherry red color represents the presence of the flavonoid. That means Shinoda test is the most unique type of test used to identify the flavonoids. So the correct answer is the C option. Next. Please the following mechanism of toxin delivery in order from most effective to least effective. We know that it is also related with the bioavailability or the effectiveness. Among the five different which has the fast bioavailability, that means fast effect. If we consider intravenous route, direct absorption, so it has maximum bioavailability. Next, inhalation, where there is a direct absorption from the alveoli. Next one, subcutaneous. Subcutaneous region is highly vascular, so it also has the good absorption. Uh, oral route also comparatively, these three oral route has less absorption, but it is better than the dermal because dermal layer is the vascular. So the correct answer is the B option, which gives the sequence of most effective, that is 1, 4, 2, 3, 5. Next, which compound is not an isomer of the other three? Uh, Here they have asked about the isomer. If you see, in case of the N-pentane, 2,2-dimethyl propane and 2-methyl butane, these three compounds are the isomer, where isomers of each other, whereas the 2,3-dimethyl butane is not the isomer. So the correct answer is the fourth option. Next, which of the following reaction of reduction is carried out in presence of hydrazones with a strong base? Uh, this particularly which reaction is carried out in presence of hydrazone along with the strong base? Okay, particularly this Wolff-Kishner reduction is the test which is carried out in presence of hydrazones and strong base, and it is used for the production of hydrocarbon from the carbonyl compound, whereas the bridge reduction is used to reduce the aromatic and non-aromatic ring. Whereas in case of the in case of the Clemson's reduction, it is used to convert the carbonyl group into the methylene group, whereas metal hydride reduction is used to reduce aldehydes and ketones to alcohol. That means about this particular statement, correct answer is the B option. Next, describe the particulars to be shown in manufacturing record. Which particular schedule gives the particulars for the manufacturing? And the correct answer is the Schedule U, whereas Schedule M gives the GMP and requirements for premises, plants, and equipment. Schedule Y gives the requirements and GMP for the requirements for the clinical trials of new drugs, whereas Schedule C is related with the biological products and their regulation. And the particulars required is mentioned in Schedule M. So the correct answer is the Schedule M. Next, which one of the following drug does not cross blood-brain barrier? We know that drugs which particularly affects on brain have the good ability to cross the blood-brain barrier. In case of the we see chloramphenicol, it crosses the blood-brain barriers. It is the antibiotic systemic circulation. In case of the sulfanilamide, it is also the antibacterial, and it is also used for the systemic circulation. It has ability to cross the blood-brain barrier. Whereas the penicillin is the broad-spectrum antibiotics, it do not crosses the blood-brain barrier. So the correct answer which do not crosses the blood barrier is the C option, that is the penicillin. Next, which one of the following is not true? We have to identify the false statement among the four statements. Drug release from reservoir system is controlled by diffusion. This is the correct statement. Drug release from matrix system is controlled by diffusion. It is also the correct statement. Drug release from reservoir systems normally follow the zero order kinetics. This is also the correct statement. Drug released from matrix system normally follow the zero order kinetic. This is the wrong statement. Actually, zero order kinetic uh, undergo constant elimination regardless of the plasma concentration. That means actually in this particular zero order kinetic, rate does not changes with an increase or decrease in the concentration of reactor. In matrix system, a release of the drug takes place by dissolution, controlled and diffusion controlled mechanism in matrix system. So particularly this statement is the wrong statement. Next, the value for dash can be obtained by comparing the rates of hydrolysis of substituted hydrolysis ester against the standard ester under acidic condition. In case of this particular, they have asked the value for dash, means particularly which particular value for dash can be obtained by comparing among the four, which particular uh, method we are using, or which by comparing the hydrogens, what can be determined? It's in very simple language. Answer is the dissociation constant, wrong one. Molar refractivity, wrong one. Uh, Taft's the steric factor, this is the correct answer. And Hammond's substituent constant, this is also wrong one. Actually, dissociation constant represents, or how it is determined, it is simply by dividing individual concentration of dissociated ion by concentration of the solution. Whereas in case of the molar refractivity, it is calculated from refractive index using the quartz corner, Lawrence constant. Whereas the Hammett substituents constant gives the degree of electron withdrawing and donating the nature. That means hydrolysis of substituted hydrolysis ester against the standard ester under acidic condition. This particular rate is given by the Taft's steric factor. Next, bioassay. Bioassay is used whenever we are doing the pharmacological screening of the drug, we mostly go for with the bioassay. Now, why the bioassay we are using? Number one, to measure the pharmacological activity of new or chemically undefined substance. Right? It is the correct one. It is also used to investigate the function of endogenous mediator. It is also correct one. It is also used to ensure the drug toxicity and unwanted effect. Right. That means bioassay is used for the identification of therapeutic effect as well as its toxicity. So the correct answer is the D option. Next, which of the following detector are widely used to detect environmental sample like chlorinated pesticides and polychlorinated bipgenes? Means which particular detector we are using for identification of the environmental sample. Uh, uh, in this, if we consider this electron capture detector, is mostly we are using to detect the environmental samples for the particularly for identification of unwanted compounds. Example is the pesticide. Whereas this thermal conductivity detector also used, but it is not for environmental. It is used to detect the gas or light hydrocarbons present in the environment. Whereas in case of the flame ionization detector, it is used to identify the vaporized gas. Whereas argon ionization detector is used for the identification of excitation of argon atoms from the radioactive substances. Environmental sample like pesticides, we are using the electron capture detector. Next, high hepatic extraction ratio is seen in all except there are the four different names of the drugs, and out of that, we have to identify the drug having the high hepatic extraction ratio. Actually, hepatic extraction ratio is the fraction of drug which is present in the liver that is eliminated during the single pass. And in case of the ampicillin, it is having the high extraction ratio. That means it is rapidly and extensively cleared from the blood by the liver. That means it is having the liver has the high clearance capacity for the ampicillin. So it is having the high hepatic extraction ratio. The correct answer is the C option, whereas propranolol, lidocaine, and theophylline has the less hepatic extraction ratio. Next, required therapeutic drug monitoring. That means which of the following drug required the therapeutic drug monitoring? Actually, therapeutic drug monitoring measures the amount of certain medicine, uh, 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 amount of medicine in your blood. That means, in general, it checks the safety and effective drug concentration in the blood. In case of the particularly which drug requires the therapeutic drug monitoring. All the three drugs, digoxin, phenytoin, and theophylline, these four drug required the drug monitoring because the digoxin is used for the cardiovascular complications, example heart failure. Phenytoin is used as an anticonvulsant in case of the seizures, whereas the theophylline is used in respiratory complications like asthma. That means all the three drugs are important and they have a very is required. Next, uh, acceptance criteria for microbiological test for herbal products falls under criteria A as per British Pharmacopoeia. Mm, acceptance criteria for microbiological test for herbal products falls under criteria A as per British Pharmacopoeia. It is the very straightforward. You should know the you should have the knowledge about the British Pharmacopoeia. Acceptance criteria is not more than 5 into 10⁻⁷ CFU per gram or per ml. Correct answer is the B option. Products are not prepared under the aseptic condition are required to be free from pathogens. The products which are not produced under the aseptic condition should do not have particularly E. coli. Example is the E. coli, and other example is
Salmonella, Pyocynia, etc. Next, are you getting my voice?
yes, continue, sir.
Yes. Next question is ISSR, a PCR-based application, is unique and inexpensive popular technique of DNA fingerprinting, which includes the characterization of, in particularly ISSR, PCR based application. Is a unique and inexpensive popular technique of DNA fingerprinting, which includes the characterization of. That means, in particular, this ISSR, that is nothing but inter simple sequence repeat polymer chain reaction based application. DNA fingerprinting, which include the character. By using this particular technique, characterization of what is done or which particular is characterized by using this particular technique? The answer is the adulteration. That means in characterization of adulteration, we are using this particular technique. But this technique is not used for heavy metal detection in DNA or determination of plant constituent. But we can identify the adulteration by using this particular technique. Next, USP apparatus 6 is USP apparatus 6 is used for testing dissolution of different dosage form. And in this particular apparatus, cylinder modified USP basket is present. So the correct answer about USP apparatus 6 is the C option. It is not related with the pedal or reciprocating disc or pedal over the disc. Correct answer is the C option. Next, question number 31. F is equal to M by M is equal to Km Tn dash model. This particular represents the Pepas and Corsin-Mayer model. Now, this Pepas-Corsin model is also called as the power law. It is used to describe and analyze the release of a drug from polymeric nanoparticle dosage forms. Example is the hydrogel. That means this particular given equation is related with the Pepas model. It is not related with the U2 model or first order or zero order. So the correct answer is the B option. Dash is the most commonly used experimental animal for modeling of the modeling human cancer. For modeling human cancer, we mostly use the mouse, whereas dog is generally we are using for the screening of pesticides or other chemical. In case of the rabbit, rabbits are used for the pyrogenicity tests of drugs and cosmetics, whereas rats are used in different drug screening, example, the cardiovascular drug. That means for modeling human cancer, mouse is mostly preferred. Next. Identified or unidentified impurities arising from the synthesis manufacturing process, such as material, intermediate, or by products, and do not increase and storage, or identified or unidentified degradation product that result from drug substances or drug product manufacturing process or arising during the storage of a material R. They have mentioned condition A and condition B, and we have to identify the type of material. It is related with the or this particular material represent the related substance. Related substance is nothing but these are the substances which are produced during this particular process. Foreign substances are the external substances. Stereometric impurities are the another kind of impurities. Toxic impurities also the same drug, but might be it also include the other drugs also or other compounds also. particularly in this condition or this statement is related with the related substances. Next, dash define two quantities to describe a degree of mixing, namely the scale of segregation and intensity of segregation. Intensity of segregation. Particularly, they have asked about the terminology and two quantities to describe degree of mixing, namely the scale of segregation and intensity of segregation. It is related with the Danvers the theory or Danvers terminology. This particular fix gives the flow of ions due to diffusion and the ion concentration gradient across the cell membrane. That means Fick's law or Fick's theory represents ion related movement. Whereas Newton is related with, or this particular Newton terminology, or it defines the force necessary to provide a mass of 1 kilogram with an acceleration of 1 meter per second. Whereas this Higuchi gives or it describes the drug release as a diffusion process such as from a matrix. That means two quantities to describe degree of mixing, namely the scale of segregation and intensity of segregation is related with the Danvers. Next, the activation energy of a reaction at a given temperature is found to be 2.303 RTJ mole. The ratio of rate constant to the Arrhenius factor is. It is related with the calculation. Here, this Arrhenius equation, the Arrhenius equation, if you put these values in the equation, the answer is 10 raised to minus 1. That is nothing but the 0.1. So the Arrhenius factor in this particular condition is the 0.1. Next, question number 36. In the GMP part 1A indicate, GMP part 1A indicate, this particularly GMP part 1A indicate the specific requirement for manufacture of sterile products, parenteral preparations, small volume injectable, large volume parenterals, and sterile ophthalmic preparations. This particular GMP of part 1A is related with these particular parenteral products. It is not related with the meta dose in inhalers or the oral liquids or oral dosage forms. Identify the name of drug. They have given the diagram or structure of drug, and we have to identify this particular structure. This structure is a bipyridine hydrochloride structure. This is the straightforward question, and you should know the structures of different drugs. Next, this particularly bipyridine hydrochloride is used for the nervous system related disorder. It improves the muscle control and reduces the stiffness. And that means it is used for the treatment of disorders such as the Parkinson's disease. Next, dash dash is a pentahydroxyflavone having 5 hydroxyl group placed at the 3 3 dash 3 comma 3 dash 4 comma 4 dash 5 dash 7 position. Simply they have mentioned the structure and they have asked about the placement of hydroxyl group at different positions. If we place the hydroxyl group at the different positions in this particular pentahydroxyflavone ring, we will get a structure of quercetin. So the correct answer is the A option, quercetin. Next, piperidine hydrocyclic ring system present in which one of the following? That means they have mentioned the food names of the different drugs, and in these drugs, which drug has the piperidine pyridine hydrocyclic ring? In case of the nicotine, if you see the structure of nicotine, here we will get the pyridine ring as well as the pyrrolidine ring. That means this ring is present in the nicotine. So the correct answer is the first option. Next, dash dash is identified and characterization of crude drug with respect to phytochemical constituent. If you want to identify the phytoconstituents or phytochemicals, we generally prefer the qualitative test of chemical evaluation, not the quantitative test. So the for qualitative determination of phytoconstituent, we use the qualitative chemical evaluation. So the correct answer is the B option, not qualitative first one. Sorry, quantitative first one. Chromatographic examination is also the wrong answer, and D, toxicological studies also the wrong answer. Next, the Hammett substituent constant H is this particularly Hammett equation is used to describe the free energy relationship in relation with reaction rate and equilibrium constant for different reactions. And this Hamilton substituent constant is zero. So the correct answer is the zero. This is the very straightforward question. You must know the constant value. Next, calculate the value of a chemical shift if observed shift from TMS is 150 mHz and operation frequency from the instrument is 100 mHz. This is the very straightforward question. You must know the if you want to calculate the chemical shift, we have to do the 150 divided by 100. The answer is the 1.5. And to that, we are multiplying with the 10 raised to minus 6. That means correct answer is the first option. If you divide 150 by 100, you will get the answer of 1.5. And in calculation for 10 raised to minus 6, that means correct answer is it is related with the calculation part, and the correct answer is the first option. Next, and anhedonia dash a general lack of interest in pleasure of life is a core of symptom of. No doubt this particular kind of characters or features is observed in disorders, particularly the behavioral disorders, and the answer is the depression. In depression, a person lack its interest from the life, which is not often observed in case of the cancer or diabetes or tuberculosis. So the correct answer is the D option. Next, hydrolysis is often the main degradation pathway for the drugs substance having dash dash functional group within their structure. That means hydrolysis generally occurs